Bismillahirrahmanirrahim. Our next topic under the unit biological molecules is we are supposed to give an account of structure and function of RNA. So let's proceed further. So this document is showing and giving us all the information related and given in our intermediate syllabus and a little bit more. So let's start with the proceedings. So RNA, ribonucleic acid, is a polymerized molecule like DNA. RNA is a polymer made up of ribonucleotides, not deoxyribonucleotides. So RNA molecules usually exist as single strand, although they can fold back on themselves, forming region with double helical characteristics. The nitrogen bases in RNA, they are cytosine, guanine, uracil, and adenine. And if there is pairing, then cytosine can combine with G and uracil with A. So RNA is synthesized from DNA through a process we know transcription. Types of RNA, there are three main types of RNA: messenger, transfer, and ribosomal RNA. All three are transcribed from DNA in the nucleus and then transported to the cytoplasm. So where they can carry out special function in protein synthesis. As we know, this is the essence of central dogma of life, that the DNA will proceed for the formation and synthesis of RNA, and then RNA, messenger RNA, and rest of the RNAs, they will take part in synthesis of proteins, and proteins are responsible for maintaining and controlling all the functions of life in cell and even organisms. So messenger RNA, it is a name suggesting its activity because it is a type of message. So messenger RNA carries genetic information from DNA in the nucleus to the ribosome in the cytoplasm where proteins are synthesized. Messenger RNA provides the template that determines the order in which amino acids are assembled to form specific proteins. It is a single-stranded molecule with variable length depending upon the type of protein they are supposed to translate. So if a protein is having 1,000 amino acids, so messenger RNA, there should be 3,000 nucleotides. If we consider the total presence of messenger RNA in a cell, so it is almost 3 to 4% of the total RNA in cell. Still, it's a variable depending upon the metabolic ability and activity of the cell. So this is general idea: DNA transcription, messenger RNA, and even rest of the RNAs transfer, ribosomal. This will be transcription, and then translation into proteins. Transfer RNA. It is about 10 to 20% of the cellular RNA. These molecules are relatively small, maybe having 75 to 90 nucleotides, depending upon different type of amino acid they carry. Transfer RNA functions by transporting specific amino acid to the ribosomes. So their function is to adopt amino acids and to take them to the metabolic, the protein synthesizing machinery, that is ribosome. So at least there should be 20 different types of amino acids. So, and they are responsible for the a bond forming between amino acid and the transfer RNA. The enzyme will take part in it. Ribosomal RNA. Ribosomal RNA is the major RNA as far as quantity is concerned. It is about 80% of the total cellular RNA and closely associated with ribosomal protein to form the ribosomes. So about 40 to 50% of the ribosome mass consists of RNA and rest is of proteins. So ribosomes serve as the site where messenger RNA and transfer RNA interact, so translating genetic information to specific protein sequence. Thus, RNA plays a crucial role in the machinery for protein synthesis. So ribonucleic acid is a vital biomolecule that plays diverse and central role in all living organisms, from carrying genetic information to catalytic biochemical reaction. We will discuss it in a brief manner shortly. Unlike its more stable cousin DNA, RNA is typically single-stranded and contain sugar ribose instead of deoxyribose and uracil instead of thymine. So, structure of RNA. This is the required portion of our syllabus. So, structure-wise, if we talk about primary structure of RNA, the primary structure of RNA is simply the linear sequence of nucleotides, ribonucleotides in it. So, nitrogen base, adenine, guanine, cytosine, uracil, 5-carbon sugar, ribose, uh, as compared to deoxyribose, where at position carbon number 2, which the oxygen is less, but in this case, oxygen is there. A phosphate group, obviously, like like the DNA structure, the phosphate group is present to form a nucleotide, and the different nucleotides, they unite together to form phosphodiester bond between carbon number 5 of one ribose and the carbon number 3 of the next. So secondary structure, although RNA is primarily single-stranded, intramolecular base pairing often occurs between complementary sequences within the same RNA molecule. This is important. There is no counterpart. There is no other chain as compared as in DNA. So this folding, this secondary structure may be of helices region where complementary bases A, U, and GC can pair up to form double helical structure or segment. Hairpin loop, the most common secondary structure where single strand loop in form is formed as the at the end of the helical stem. So once again, pairing will be of same. There are some bulges regions where one strand of helix is having extra nucleotide, so there will be no counterpart, and that will form a bulge. So internal loops, small series of unpaired bases with a longer paired helix. So pseudonodes, most complex structure form within a hairpin loop itself form base pair with sequences outside of its own stem, creating a knot-like arrangement. So this is a sort of sketch showing the four types of interaction: single strand, bulges where there is no counterpart, and internal loops. It is forming a sort of loop because there was no what we say A can't interlink with the other A, so there will be a bulge in that. So hairpin-like structure in which there is no bonding, there is no interlinks, but these structure can form. So this is hairpin-like structure. This is secondary structure. What about tertiary structure? Tertiary structure refers to the precise three-dimensional arrangement of RNA molecules in space. This is achieved through further folding and interaction between secondary structural elements. These interactions are stabilized by various forces like base stacking, hydrophobic interaction between adjacent bases, hydrogen bonds beyond canonical AU and GC pairs, non-canonical wobble and Hochstein base pairs can form, particularly in RNA, contributing to complex tertiary structures. Electrostatic interactions, where there are some charges, so they can form interactions. So specific motifs, these are special structure which can act as a what we say, they can play metabolic role as well. So the specific motifs, they are there. I'll see I'll discuss it later on. So in this case, if we study the tertiary structure, so this was the primary structure. Primary structure, RNA sequence responsible for the presence of codon on it. Then secondary structure, there will be binding and hairpin-like structure or bulges. So in case of tertiary structure, there are knots, multiple secondary structures they form higher order tertiary bonds. Motifs can function as catalysts. So these are the tertiary structures. You know, these knots, they are not of primary nature, but there will be bonding between them. So they may be more stable, and they can perform certain catalytic functions. So functions of RNA: protein synthesis due to central dogma of life function, messenger RNA carrying the message, having codons, ribosomal RNA providing scaffold, providing ribosomes for the interaction of messenger RNA, ribosomal RNA carrying amino acid on them. So ribosomal RNA plays very important role because they are the structure responsible for the formation of ribosomes. So transfer RNA carrying the required amino acid. So other than these functions, they may work as genetic material in viruses. For example, some viruses are having RNA as their primary genetic material instead of DNA, like HIV, influenza virus. And then gene function role. Many types of non-coding RNAs, ncRNAs, play a critical role in regulating gene expression as at various levels. And what are these? These are given in a short manner. Micro RNAs, abbreviated as miRNAs, small single-stranded RNA that bind to complementary messenger RNA sequence, leading to inhibition of translation or degradation of the messenger RNA. Their function is to control the silencing of the gene if it is required. Small interfering RNAs, siRNAs, similar to miRNAs, but siRNAs are typically derived from exogenous sources like viruses or transposons, and they mediate RNA interference like the previous one, and leading to the degradation of target RNA. And they help to protect the cell by guiding protein to destroy the matching messenger RNA molecules, or stopping the harmful genes which may be present within the cell, or viruses which carries those those genes. So long non-coding RNAs, a diverse group of RNA molecule longer than 200 nucleotides that regulate gene expression through various mechanisms, including epigenetic modification, transcription control, and post-transcriptional processing. Then comes the ribo switches, regulatory RNA elements often found in untranslated region of the messenger RNA that directly bind small molecules, metabolites, and undergo conformational changes to regulate the gene expression and control of transcription or translation factors. So catalytic activity, ribozymes, this is well-known example. So some RNA molecules possess enzymatic activity acting as biological catalysts. These are called as ribozymes. So there are some examples like ribosomal RNA. Sometimes it may work as ribozyme. The peptidyl transferase activity of the ribosome responsible for forming peptide bond during protein synthesis is catalyzed by ribosomal RNA. So spliceosomal RNA involved in splicing of pre-messenger RNA to remove introns, self-splicing introns, certain RNA molecules that can excise themselves from a larger RNA transcript without the need for protein enzymes. So they will act by themselves. So they are there. So telomeric maintenance, telomerase, an enzyme that maintain the ends of chromosome telomers. We know the size become lesser and lesser due to the repeated cell division, but the enzyme responsible to add on more nucleotides. So contain an RNA component that serve as template for adding telomeric repeats. RNA processing and modification, small nuclear RNAs, snRNAs, and small nuclear RNAs, SNOs, nucleolar RNAs, SNORNAs are involved in processing, modification, and assembly of other RNA molecules, particularly ribosomal and transfer RNA. So different RNAs can also control the synthesis and modification of transfer and ribosomal RNA, and they are important because they play their important role in the modification of these major RNAs taking part in protein synthesis. So, because RNA can store information as well as catalytic function, most scientists believe that there was RNA world hypothesis in the scientific community, and this is the strong explanation of how the life began, because they thought that RNA was acting before the assembly and before the intervention of DNA and even proteins. So in summary, RNA is highly dynamic and versatile molecule with a complex structure that enables it to perform a vast array of essential functions, from the fundamental processing of protein synthesis to intricate gene regulation and even enzymatic activity. So this is all about the structure and function of RNAs given in our syllabus, although the last part may be not required, but we are covering it because it is a short document and we can easily understand and grasp the concept given it. So next time I will discuss the conjugated molecules. Till then, Allah Hafiz.